Hello, in today's video, we shall learn in detail about glycolysis, which is a primary step of cellular respiration. By the end of this video, you'll have a clear understanding of the two major phases of glycolysis, namely the preparatory phase and the payoff phase. Let us start with the introduction to glycolysis. Glycolysis is a biochemical pathway in which glucose is broken down to produce energy in the form of adenosine triphosphate. It produces two molecules of pyruvate, adenosine triphosphate, and nicotinamide adenine dinucleotide. Glycolysis takes place in the cytoplasm of all living cells, aerobic as well as anaerobic, and does not require oxygen. Glycolysis is the primary step of cellular respiration. It is also known as Embden-Meyerhof-Parnas pathway or EMP pathway. Glycolysis consists of two major phases. First phase is the preparatory phase consisting of 5 steps. It involves phosphorylation of glucose, conversion to glyceraldehyde 3-phosphate at the expense of 2 molecules of ATP. Second phase is the oxidative and payoff phase. It involves conversion of glyceraldehyde 3-phosphate to pyruvate and formation of ATP and NADH2. Let us take an account of the preparatory phase. First step is phosphorylation. 6-carbon glucose molecule undergoes phosphorylation with the help of ATP. It is catalyzed by enzyme hexokinase and the cofactor magnesium ions and yields glucose 6-phosphate, a 6-carbon compound, and ADP. Second step is isomerization. Glucose 6-phosphate is converted into its isomer fructose 6-phosphate in the presence of phosphoglucoisomerase enzyme. Third step is phosphorylation. Fructose 6-phosphate is further phosphorylated by ATP molecule to give fructose 1,6-bisphosphate and ADP. This reaction is catalyzed by the enzyme phosphofructokinase and magnesium ions. The fourth phase is cleavage. Enzyme fructose 1,6-bisphosphate aldolase catalyzes the cleavage or splitting of fructose 1,6-bisphosphate to yield 2 different triose-sugar phosphates, namely glyceraldehyde 3-phosphate and dihydroxyacetone phosphate or DHAP. Fifth step is isomerization. The triose phosphates, namely glyceraldehyde 3-phosphate and hydroxyacetone phosphate, are isomers and are interconvertible. DHAP is converted to glyceraldehyde 3-phosphate by enzyme phosphotriose isomerase. Thus, in the initial phase of glycolysis or in the preparatory phase, Glucose molecule is activated by phosphorylation and then splits into two molecules of triose phosphates, namely glyceraldehyde 3-phosphate and DHAP. Glyceraldehyde 3-phosphate takes part in the formation of pyruvate, while DHAP gets converted into glyceraldehyde 3-phosphate. Therefore, two molecules of PGAL or glyceraldehyde 3-phosphate are formed. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Next, let us learn about the payoff phase. The sixth step is oxidation and phosphorylation. In the presence of enzyme phosphoglyceraldehyde dehydrogenase, glyceraldehyde 3-phosphate is oxidized to 1,3-bisphosphoglycerate. During this reaction, NAD takes up 2 hydrogen ions and gets reduced to NADH2. Therefore, in this step, both dehydrogenation and phosphorylation takes place. Next step is ATP generation. 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate by the removal of a phosphate group. This phosphate combines with ADP to form an ATP molecule in the presence of enzyme phosphoglycerate kinase. This is followed by isomerization. 3-phosphoglycerate is converted into its isomer 2-phosphoglycerate with the help of enzyme phosphoglycerate mutase. Next step is dehydration. 2-phosphoglycerate undergoes dehydration in the presence of enzyme enolase and cofactor magnesium ions to form phosphoenol pyruvate. One water molecule is released. The final step is ATP generation. On hydrolysis, phosphoenol pyruvate loses a phosphate group and forms pyruvate. The released phosphate group combines with ADP to generate ATP in the presence of enzyme pyruvate kinase. At the end of glycolysis, one glucose molecule is converted into 2 molecules of pyruvate. The overall equation for glycolysis may be written as shown. The first 5 steps of EMP pathway or glycolysis is known as preparatory phase. Energy is consumed to produce 2 molecules of triose sugar phosphate. The next 5 steps of EMP pathway is known as the payoff phase. It forms pyruvate, ATP, and nicotinamide adenine dinucleotide molecules. So today, we have learnt in detail about glycolysis under 2 separate headings, namely preparatory phase and payoff phase, with the help of equations in stepwise manner. I have some practice questions for you. Please share your answers in the comments section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.